Friends, good evening. It is an enormous night for DRIFCan, for diabetes research, as we look to raise funds and accelerate a cure for diabetes. We're beginning our broadcast a little bit early because I see many of you have already logged on and are waiting for our live broadcast to begin. And as you do so, I want to encourage you to introduce yourself in the comments section here on our technology that we're using tonight. It's called StreamYard. You can post a comment throughout the evening. You can post questions. And really what this does is it creates community tonight as we are raising money for DRIFCans and move toward a cure for diabetes. We are so close, and you're going to hear more about that tonight. But we welcome your comments. And more importantly, there are so many of you that work in this field. There are diabetic patients joining us, families, people from around the globe. Go ahead and introduce yourself and let me know where you're joining us from, because I'm excited to share it with the rest of our viewers, because I know many of you that are watching this broadcast likely know each other. So I encourage you at this time, introduce yourself. Let me know where it is you're joining us from, where you're watching, why you're watching, and I will read your comments and your questions as they come in real time. I love and I appreciate the conversation, and I know Dr. Shapiro appreciates the questions as well. So all of you are being far too quiet right now. There we go. Kathy Westling, Kathy from Breton, Alberta. Grandma to a type 1 diabetic. Welcome, Kathy. Thank you for introducing yourself. Just like Kathy did, go ahead. Hello to you, Joel, a father of a wonderful 9-year-old from Calgary, of a type 1 diabetic. Thank you so much, Joel, for being with us this evening. So I expect to see many of you. Maureen B. from Edmonton. My adult daughter is type 1 diagnosed at age 7. Maureen, tonight is going to be an exciting learning journey for you as well as everybody else who is watching tonight. Thanks for saying hello. I appreciate it. Philip Schaefer, hello to you. He says, hi, Carrie. We're from Richmond Hill, Ontario. Our 3-year-old toddler son has type 1 diabetes. Thanks for saying hello, Philip. Jim and Tanya. Oh, I jumped ahead here. Jim and Tanya Jankovic says, hi, Carrie. I'm Jim from Windsor, Ontario, type 1 diabetic for 44 years. Welcome to both of you. Hello to Faye Evans, mother of a type 1 diabetic from Leduc, Alberta. Hello to you as well, Faye. Joel Shusette, I believe, says, oh, they're coming in fast and furious now, hoping my alma mater can be a huge part of the solution to curing type 1 diabetes. I see a few people in the green room nodding. I think they know you as well. I'm going to keep clicking through here because now everybody is saying hello. We have a registered dietitian, Marcy, joining us from Oregon, United States. Hello to you. Tori Oliver from Lloydminster, mom to an 11-year-old. Catherine Jeans from Calgary, type 1 for 32 years. My goodness. Sherry Simpson, type 1 diabetic for 47 years. Carly says, hello, my 12-year-old son Cash was diagnosed just this year. We have Burgundy from Red Lake, Ontario, mom to a type 1 diabetic, 8 years old. Bethany Rose is a type 1 diabetic for 40 years. So excited to hear from Dr. Shapiro tonight. Oh my goodness, I love all of these comments coming in. Paul and Joanne, type 1 diabetic, been part of the transplant program since 2009. Paige says, hello from Sylvan Lake, type 1 diabetic for about 16 years. So many of you rather, joining us from all across Alberta and all across Canada and the United States. Silver Moon Music says, hello, my name is Joanne Brown, 43-year-old male from Edmonton, type 1 at the age of 27. I'm going to throw this out there right now as all of you are introducing yourselves. Dr. Shapiro needs patience for his trials. We are going to talk about that tonight several times. So I'm going to plant the seed right before we get going and let all of you know that if you feel up to participating. This is how we move forward in our step towards a cure, our step towards new treatments. We need you. So if you are willing, please, we need you to sign up, and I will give you the email and where to do that momentarily. So stand by for that. But as you are listening and watching tonight, I encourage you to think about participating, because that's how Dr. Shapiro and his unbelievable team based in Edmonton can understand if the treatments are working and move forward and make progress. In the interest of time, I love all of the comments that are coming in. Please continue to introduce yourself. Let us know where you're from. And as I move through the program tonight, I encourage you to join in on this conversation. It creates community. It allows each of you to know where other people are joining from and where they're going through. And also you can bring forward your questions, and I can relate them to Dr. Shapiro in real time. This is big, and we appreciate your time. So on that note, I am going to begin and say, ladies and gentlemen, researchers and scientists, patients, family, and friends joining us from around the globe, hello, and welcome to our second annual virtual fundraiser, Accelerate a Cure for Diabetes, with world-renowned diabetes researcher, Dr. James Shapiro, along with two of his patients. And it's all in support of the Diabetes Research Institute Foundation of Canada, more commonly known as, and I will refer as, DRIFCan. My name is Carrie Dahl. I was a longtime television news anchor and medical reporter turned podcast host and public speaking coach. And I am so honored to serve as your MC for tonight's virtual event. We do appreciate all the time that you are taking out of your lives to spend with us tonight. And of course, to learn more about DRIFCan, along with what Dr. Shapiro is up to. And then, of course, the big goal tonight is to raise important funds for his research to accelerate a cure for diabetes. I want to begin early on by thanking our event sponsor, Leading Edge Physiotherapy. Thank you so much to the team there. We appreciate you, and we appreciate the fact that you stepped forward. You're champions in our community. You help make events like this happen. So to the entire team at Leading Edge Physiotherapy, thank you so much for your sponsorship tonight. As I mentioned, we want this event to be as informative and as interactive as possible. So please join in this conversation. You asking questions in real time is what makes this event unique. You can type them in the chat box at any time. I will read them and weave them into the conversation. Thank you for participating. On that note, this just popped up. I wonder if Dr. Shapiro knows them. It's DZIF, listening from Leeds, United Kingdom. How exciting is that? I can see Dr. Shapiro right now in the virtual green room nodding his head. I think the two of you might know each other. So there we are, listeners and viewers from around the world. Thank you, Dee, for saying hello tonight. The format is simple this evening. Over the next 90 minutes, I will begin by giving you some background about DRIFCan and explain why tonight is such an important fundraiser. I want to stress very early on tonight that when you make a donation, no matter where you are joining us from, even if it is from the United Kingdom, your donation will and can be made in the denomination of the country that you live in. So whatever dollars you want to donate in, you can do that tonight. And all you have to do is go to DRIFCan.com. That is where you can make the donation tonight. I will be repeating that website many, many times throughout the evening, DRIFCan.com. That's where you make the donations tonight in the denomination of where you live in. And here is the big bonus, everybody. You will get a tax receipt for your donation tonight. So please do keep that in mind. So momentarily, I'm going to begin by doing an interview with Dr. James Shapiro. I know so many of you are looking forward to hearing from him, what he's up to, and of course, asking questions to him. He's going to talk about his work and the exciting step forward that him and his team are taking. Plus, we have a really big surprise for those of you watching tonight. So stay tuned for that. and of course, welcoming your patients or welcoming your questions, pardon me, along the way. Then you're going to hear from two of Dr. Shapiro's patients. Kathleen Gibson underwent 3 islet cell transplants, and she will talk about the impact that that has had on her life. Kerry Elliott will also be joining us. He is the first patient in Canada to undergo the VioCyte stem cell transplant. This was and is a huge step forward in treatment and research, and Kerry will tell us more about his experience. Then Rob Otway, the chair of the DRIFCan board, will join us a little bit later to explain how the funds raised tonight are used by Dr. Shapiro and his team. He will explain the future goals for the organization as well. And then we're going to wrap up the evening at 7:30 p.m. Mountain Standard Time. So with that, let's begin with a little more information about DRIFCan. Their mission, laser focused on accelerating the cure for diabetes by directly funding diabetes researcher Dr. James Shapiro and his team at the University of Alberta. November is Diabetes Awareness Month, and you are encouraged, if you are a supporter or a patient, to wear blue. That's why I have blue on tonight, and you will notice that all of the guests joining us this evening are wearing blue as well. And today we are celebrating as well. I'm not going to give the details on that, but Dr. Shapiro will allude to why today is such a special anniversary. And it is huge in the world of diabetes. Also, a few numbers I want you all to think about tonight and to digest. There are over 450 million people around the world with a form of diabetes. About 10% of them have type 1 diabetes. The current cost of diabetes to our healthcare system, you ready for this? $14 billion a year. $14 billion is what diabetes costs our healthcare system. Now, just imagine if we could take some of that $14 billion and invest it in Dr. Shapiro's work. Wouldn't that be exciting and taking step forwards to accelerating the mission towards a cure? So Dr. Shapiro's research and his amazing team is changing and impacting countless lives around the world. That said, funding is crucial to its success. And with that in mind, I want to remind all of you tonight that the reason we are gathering is to raise funds for DRIFCan. As I just mentioned, the government does not fund this type of research. So that's why fundraising like what we are doing tonight is crucial to see the advances continue to move forward toward a cure. And I have some really exciting news to share with you right now, and I'm going to do it early on so that we can maximize the benefits. You ready for this? An anonymous donor has come forward and said that they will match up to $500,000 in donations during the month of November. This is huge for the organization, for DRIFCan. So keep in mind tonight that whatever you are able to donate will essentially be doubled, which is super exciting because that is impact. And all the money tonight goes directly to fund the research of Dr. Shapiro and his amazing team. How can you make the donations? Again, go to the website, DRIFCan.com. It is super easy. Just follow the steps. And as I mentioned, you can make the donation in the denomination of where you live. One more time again, I want to say a big thank you to our sponsor tonight, Leading Edge Physiotherapy. They have come on board and helped us out with this event. And once again, to all of you, we appreciate you. So without further ado, it is now my pleasure to introduce our special guest tonight, Dr. James Shapiro. James Shapiro was born in Leeds, England. He's the son of a family doctor. He studied medicine in Newcastle, trained in surgery in Bristol, and developed an interest in islet transplantation as a medical student. He has been on the faculty of the University of Alberta since 1998, where he now holds the Canada Research Chair in Transplantation Surgery and Regenerative Medicine. He directs the Living Donor Liver Transplant and the islet transplant programs in Edmonton. He was the lead investigator on the famous Edmonton Protocol cell transplant treatment for diabetes. He is the Project 1 lead for the CNTRP, which is actively researching ex vivo organ transplant repair. In 2015, his team began the first in-human trials with embryonic stem cell-derived insulin-producing cells transplanted in an immuno-isolation device. Over the years, friends, as I was a television news anchor and a medical reporter, I had the honor, and it really was always an honor and the opportunity to interview Dr. Shapiro many times because of the incredible work he was doing and the medical breakthroughs. And I know one day soon, and I say this with so much confidence, because of his work and his team's work and the medical breakthroughs, I know one day soon that we are going to be sharing with the world that they have found a cure for diabetes. I know it. I'm manifesting it now. It's going to happen in our lifetime. On that note, it is an honor and a pleasure to introduce the amazing Dr. James Shapiro, joining us from his home tonight in Edmonton, Alberta. It's so nice to see you.
You too, Carrie. Good evening, everybody. And hi, Dee in England. That's my sister, by the way. And I'm surprised.
Is it?
I'm surprised she's up so late and that she knew about this. But there you go.
Oh yeah, it's awfully late there.
Yeah.
So she's staying up late to cheer you on tonight.
Yeah,
That's wonderful.
That way, thanks. Yeah.
Dr. Shapiro, what an amazing night. And I mentioned a little bit earlier that we are celebrating a special anniversary tonight. So I said I didn't want to steal your thunder. I'm going to let you go ahead and talk about what the anniversary is today. And we have a picture to share with everybody.
Yeah, flicking through Banting and Best's notebook 100 years ago, 12 hours from now. So that was a few days ago, October the 26th, and that is November the 4th. Tomorrow morning, you can see those three numbers at the bottom right-hand side of that handwritten piece of paper by Banting. So 0.33, 0.14, 0.13. That's the first time that blood sugar fell in the animal models that they were testing with their insulin extract. And so we do celebrate a Canadian discovery insulin 100 years on tomorrow morning.
And I love that you mentioned Canadian discovery. We're keeping it in the family, aren't we, Dr. Shapiro?
Well, it's a big team. And the world is smaller today in some ways. There's more people, but we're so much more interconnected, particularly in science. We really do talk to each other. We don't have to send letters anymore. We can pick up the phone. We can have lab meetings by Zoom. Everything's changed.
It really has. And it, like you said, has made the world much smaller and increased the ability to collaborate. And on that note, though, as we look at this piece of paper and talk about it, clearly we know that that wasn't a cure. And that's why you and your team have been working around the clock for a number of years now.
Yeah. So, insulin is an amazing treatment. It saves lives, but it doesn't prevent all those awful secondary complications of the disease that patients have to face over the course of time. First of all, it's a big deal to have to inject insulin and test your blood sugar every day, multiple times. Big deal. I can't imagine it. I've never lived with it. Having a child that has it, I just can't imagine what you people tonight know far more about this disease and living it every day than I do. I just know that this injection of insulin, a drop under the skin, isn't enough. It's not good enough. It's not perfect. When we transplant cells into patients that have diabetes, we can correct the defect that's causing the diabetes in the first place. So we lose the ability to make insulin when someone gets diabetes. And the insulin is really only a Band-Aid fix. It's not a permanent cure. To be able to cure this disease, what we need to do is to restore, repair, replace the cells that get damaged inside the pancreas, those islets of Langerhans that make the insulin and release it so perfectly. This, as you can see in the x-ray there, that x-ray picture on the right-hand side, the insulin just sits there under the skin and it's absorbed slowly or rapidly, but really imprecisely. And that's not good enough. We can do
Dr. Shapiro, I want to emphasize to everybody watching that this has been an interest and really a passion of yours for many, many years. So I have to share this picture with everybody watching because you started at a very young age.
I did indeed. Yeah. Oh, yeah, that one. Okay, yeah. So that was me as a medical student in 1983, when I used to pay for my can of beans at nighttime by working for a local radio station, Radio Tyneside. But during the daytime, that's when I first developed an interest in islet cell transplantation, and worked with John Farndon in Newcastle, and for the first time began experiments. And most of those experiments for me didn't work out. It was I worked day and night because I thought I'd never get a job in surgery if I didn't do that. But that experience left me with this is a possibility. This has to happen. We need to make cell transplants work in patients.
And that brings us to, I think, a picture from you doing an islet cell transplant a number of years ago. That's where this is from, right?
That's patient number 100. So we've done 706 or so, maybe more now. It's hard to keep track of these intraportal islet transplants. Well, what that means is we're dripping those cells into the liver, inside the liver in a patient, and it doesn't require surgery to do that. The patient there is actually wide awake, and we can chat to her as we're infusing the cells. It's a simple procedure. It's done by local anesthetic on the side, freezing like you might get at the dentist, and then a little tube is threaded through into the vein inside the liver, and then we drip the cells in there, and it's a pretty good place for cells to survive. Not perfect, but pretty good.
I know how proud and thankful you are to the team that works with you and alongside you. We have a few pictures that we want to share with everybody for them to understand just how intense this process is. Explain what we're seeing here.
Well, those are actually the first patients on the top left. In fact, the very first patient on the top left there, it has been insulin-free after his islet cell transplant for 22 years. That's a world record. Completely insulin-free, normal blood sugar control, perfect for 22 years. It's we wish every patient would end up that way. We're learning more every day about why that is, but it's possible. And I think that patient shows us what the potential of this treatment really is. And then you can see that these are, as we go on, there's next is 7 patients, next is 50 patients, next is 100 patients. So we're celebrating, this growing team of patients that participate in our trials.
There are so many. And so from there, we move into your team that supports you.
Okay, so what you can see there is the lab team. So the tantalizing next steps are locked in the lab. And what we have to do is figure out how to make that work and move it as fast as we can to patients. And you can see that team there is an incredible team. We've got literally 20, 30, 40 different scientists that have worked with us over the years, but that's our current team. And you're going to meet one of those tonight. Nadish, Dr. Nadish is on the second from left there with the glasses on, and he'll be able to tell you a little bit about what our team does.
Well, on that note, we spilled the beans, so why don't we bring in Nadish right now. Hello to you, Nadish. It is so great to have you with us. And on that note, it's not just Nadish, it's Faye. And we also have, check this out, everybody, the clinical islet transplant research team coming to you live from the University of Alberta. Go ahead, Dr. Shapiro, and do the introductions, because we were looking at pictures, and we can share pictures all night, but why don't we bring in the team and talk about them?
Yeah, so I'm often put in the front of the queue, but it's actually this team that does the hard work all behind the scenes, growing the cells, making the research happen, writing the big grants to try to get approval for patients to be treated so we can try and raise funds. And so as we move to the right, you can see Dr. Nadish there. He's a senior research associate in the lab. He's got magic fingers. He's really has, and he's also able to conduct a really great team of additional scientists that work with us in the lab. So I'm thrilled to have him and thrilled with the lab team. On the bottom left there, you can see, well, it's a little small to me, but I can see Paris 2 and Rainer and Omar and Daini and I'm going to miss some Indri. And have I missed anyone off? Paris 2? Did I say Paris 2? Yeah. So awesome team. That's the clinical research team who helps find us patients for research trials, make sure that everything's approved and ready to go, and we have ethics approval and Health Canada approval. It's really enabling. Without this team of people, we couldn't do the clinical trials, the cutting edge trials that we do every day. And Faye on the right-hand side there, hi Faye. So Faye really helps to keep this team together, and also is involved with the big grants and applications and approvals. So without this team doing what they do every day, I certainly couldn't achieve what our team does.
Incredible. I want to say thank you to all of you staying late to work tonight to say hello. And while I have you all on the screen, I have some incredible news to share with you. And I thought I would keep you on this to share this news. Do you know that Will Hergott stated that he is going to donate $25,000? $25,000 to DRIFCan.
Wow. Well, thank you very much. That's amazing. Thank you.
Will, thank you so much. There we go. Up and up we go towards, like I said, $500,000 if we raise that tonight. An anonymous donor will match that as well. So we're starting out strong tonight. And I wanted to share that news with the entire team, or at least those of you who are able to join us tonight. So to all of you with us tonight, I'm going to let the clinical islet transplant research team, a number of you standing there, thank you so much. I think you either have homes to get to or more work to do. So I'm going to let you and Faye go, and I'm going to keep Nadish around because Nadish has something really exciting. So thanks to all of you for joining us. We appreciate you being with us. And Faye, thanks to you as well for everything you do. We appreciate everything that you have contributed.
Thank you, Carrie.
Nadish, you have a special treat for us tonight, and Dr. Shapiro wanted you to come forward because so often we talk about his work and your work as a team and what you're up to, but a very rare treat tonight. You are going to take us inside the lab, and this is extraordinary because not anybody, you just can't get in there unless you're part of the team, because I'm sure it's highly controlled. And tell us why, Nadish, why not just anybody can walk through those doors.
Absolutely. First of all, thank you, Carrie, for such a wonderful opportunity, and thank all the donors. So we are basically making patient-owned insulin-producing cells from stem cells, which are known as iPS cells. And the benefit of this is they are accepted from patient-own immune system, not get rejected. So this is the process of manufacturing these cells is very challenging. It has a very stringent conditions. So we have to make sure that these are safe. They should not have interference from outside environment, and we can produce most consistent and functional cells in the lab.
You are going to take us on a tour tonight, an unprecedented tour, so everybody has an idea of where Dr. Shapiro and you and your team work and what the equipment looks like. So I'm going to let you get started on that tour, and as you get up, I want to read a few comments coming in, and here they are. Rocco says, great news, Will. This is in conjunction with the $25,000 donation. C. Gale says, William, you're the man. Nicole Anne-Marie Rodriguez says, yay, wow, incredible. Shelley says, thank you, Dr. Hergott. Thank you to you, Shelley. David says, William, with a thumbs up. Lachlan says, great question, Lee. Would love to hear them discuss similarities and differences with Vertex. We're going to get to that as well. I think there's a lot of different discussions that are already going on with those that are watching. We'll get to more of those comments momentarily. Nadish is now making his way down a hallway. So Nadish, you've come to the doors. Go ahead and take it from here.
So this is the new lab where the live action is going on, and I'm so happy to share you that that's where things are happening. You can see the team is working right there. These are the cell processing experts who are making cells in those machines right there. I'm gonna give you a closer look. What are they doing here? And basically, the cells are grown in here in these little machines. These are the home for these cells, and they have a specialized apparatus which is called bioreactors. And as you can see here, the cells grows in these little reactors and produce insulin-producing beta cells.
Nadish, I have to hop in, and you can hop in as well. Dr. Shapiro, were you talking about stem cells right now?
So what we're talking about is this is a drop of blood that's taken from a patient, a tube of blood. from the forearm. And we take the blood cells, and then we can manipulate them and turn them back into any cell we want in the body. And of course, what we want for diabetes is insulin-producing cells, so that we can put them back in. Why do we want patients' own blood? The reason we want that is it's exactly compatible with them. So right now we have to use anti-rejection drugs when we transplant islet cells, and these have potentially serious side effects, increased risk of certain cancers. They can cause infections and trigger infections. And so if you had your own cells put back in, you wouldn't need any of those. So that's what we're doing here. It's a really challenging but totally exciting opportunity to make this happen with patients' own cells. And I'm really confident at the end of the day that when we develop and perfect this approach, we will have a treatment that will get as close to a cure as possible for patients with all forms of diabetes, not just type 1, but all forms of diabetes.
So exciting. Okay, Nadish, where are you now?
So I'm in a very specialized room. It's called microscopy room, and it's a very sophisticated instrument to look the stem cell derived cells that we transplant in mice, and it shows that how they look like, and they're producing beta cells over here. You can see in the green color, these are the beta cells that are implanted in the mice, and they are showing that how they function and mature. So it's a very cool technique. It's an instrument called microscope, and showing a confirmation that these cells are able to produce insulin and functional in rodents.
Dr. Shapiro, do you want to weigh in at all on that?
Well, I want to say that picture gives me goosebumps, because that is an islet cell that's been made from somebody's blood. And if you put it back into them, your body won't reject it, and it will make insulin. The green there, lots of green there, is all insulin producing cells. And that just, when I lie awake at night and think about that and look at that picture, I realize that this is actually possible.
Absolutely. Here we have a comment here that says, Lachlan asked, did he say they use the patient's own stem cells? Maybe if that's the case, no need for immunosuppressants. Really cool to see the lab. Go ahead, Dr. Shapiro.
That's exactly what we said. Yeah. So we're taking patients' blood cells, turning them, winding them back, and then winding them forward again into the cells that we want. And that's what Nadish and the team's skill is.
And Shelley says that is amazing. No rejection. You're right, Shelley. You're right. Okay, Nadish, where are you off to now?
I'm just heading back to my office again.
So that is the full tour of the lab.
So we should tell you, in this same building on the top floor there, a colleague of ours, Greg Corbett, who I've worked with for many, many years, he has an ultra clean room facility for manufacturing cells that are fit to go into patients. And so what we're doing right now is mapping our protocols and making them work in our hands, and training the team that will actually make cells for patients in the ultra clean room facility. And so that's exactly the process we're going through right now. And I'm hopeful with this approach that we'll be able to transplant our first patients with their own cells probably within a year from now.
Before I continue down that road, Nadish, I know you're finished your tour. Thank you very much. So many comments coming in saying that people loved it. As Simone says, truly amazing work. Thank you so much. I have countless comments of people saying, very exciting to see the lab. Final question to you, Nadish. What is it like working on a project like this with Dr. Shapiro?
All I would say is, I think it's a game changer. We are working on a mission, and I think the only thing that I would say is we are very close to make a day when we say that diabetes is a history, on Dr. Shapiro's word.
Incredible. Nadish, thank you so much for all the efforts that you went through. It was a bit of a technical feat that we had to conquer to be able to bring our viewers along and show us the lab, but
Absolutely.
You did it for us, and we appreciate it, and the team that stuck around to show us what they're up to. So thank you so much for joining us on this podcast and for that brilliant tour. We wouldn't have been able to see it without you.
Likewise, it's a pleasure. Thanks for this opportunity. Thank you so much.
Thank you, Nadish. Wow, that is just so exciting to be able to go through that and to see what you're up to, Dr. Shapiro. But I want to even move this a little bit forward, because now we're going to talk about fundraising and the dream. And this is part of the dream. So tell us what we're looking at here.
Okay, if you click play, there's a little video behind that. It may or may not play at your end, but that is a cocoon. And what we're trying to do here with this system, it's made by a company called Lonza. And what we're trying to do is make machines grow these cells. You know, right now our technicians, as you've seen, have to work day and night to add growth factors, change the media, and coax these cells into growing. And basically what we're doing there is making the cells think that they're inside the body, growing and developing inside an embryo. Of course, they're not. We're just making that artificially so. But what these machines will do is automate that process and make it simpler, so that once we've treated the first handful of patients, with these kind of technologies, robots and these kind of cocoons and the orchard of rooms that we can generate the cells in, we will be able to make cells eventually for thousands upon thousands of patients, hopefully one day 463 million people with all forms of diabetes across the world. That's the goal.
That's incredible. But I can imagine that equipment like this is very expensive. Can you give us an idea of what something like this would cost? Because you don't need just one, you need a room full of these, right?
Yeah, exactly. So you can see here that this is the most expensive project, unfortunately, I've ever worked on. It may be the most ambitious, but it's also the most expensive. And, you know, it costs us this kind of money every month because we are adding growth factors. They're very expensive. These media that are required to grow these cells is very expensive. And of course, our team has expanded considerably compared to where we were, 15, 20 years ago. So this is when I, as I put my head on the pillow at night, these are the things I worry about. And I know that I've got enough money right now to last me just under a year. And then this team, this really talented team of scientists, I won't be able to sustain them, and we'll have to let them go. And I just don't want to see that happen. But more importantly, I want to see a cure found from this approach. And I think working together to make this happen, we need funding. Like that's, I wish we didn't. It's not nice to talk about that, but we can't achieve what we're achieving now, and we won't be able to deliver this into patients unless we have additional funding, and we're desperately short of funding right now.
On that note, talk about, and I love being able to ask this question, how close are you to a cure?
This idea of making patients' own cells, testing it first in patients, and then getting to the point where we can deliver a full dose of cells so that a patient no longer has to take insulin and doesn't have to take any anti-rejection drugs, that is as close as a cure that we will ever get to, I think, in our lifetimes. And what I focus on, we've last 20 years with these 706 islet infusions into the liver have all been in adult patients requiring very powerful anti-rejection drugs. I long to be able to treat a child with just diagnosed with diabetes and put cells into them and have no side effects from any drugs and no long-term lifetime risk, and eliminate the diabetes, a cure essentially at the time they're diagnosed. And I think that is possible with this kind of approach. We've got a lot to prove between now and the point that we get there. But I think without getting on that journey and making it happen, we'll get nowhere. So we just have to move forward.
I have a number of comments that are coming in and questions. And Bonilla says, amazing work. I'm in tears. Hearing that you were so close to finding a cure makes me more hopeful than ever. This is as real as it gets. And you are that close. And you know what? You should be in tears because the work that's being done is incredible. Another comment from Donna says, how close are you to human trials? So maybe you want to recap that and talk about that.
Yeah, so we mentioned at the beginning that we are looking for patients actively right now. We have a number of different trials that move forward. We probably run 15 or 20 different clinical trials in patients every year to try and move this field forward, not just on the patient's own stem cell work that we're talking about, but other stem cells as well. So we have a big trial that's moving forward now, a really exciting trial that comes from a company called ViaCyte in San Diego. And what they've done is taken stem cells similar to the ones that we're making, but they're not quite the same. And those ones they have genetically altered by editing the genes using a special technology that's only recently become available. It's called CRISPR-Cas9, but the name doesn't matter. What that does is basically make the cells immunologically silent. And what do I mean by that? It means that these cells can be transplanted under the radar of the immune system. They won't be seen. They're stealthy cells. And we are hopeful that in that first approach in patients that we'll be able to transplant some of these stem cells in the next few months. We're hoping to do our first patient by December. And we hope that we'll learn from that the cells can survive without any anti-rejection drugs. It's an experiment, but it's a first in human trial and a really exciting one. We've been waiting for that kind of day, I would say, the last 20 or 30 years.
Which is so huge. But again, you need patience. And that's why I have this email up. So talk about that, the importance of having people come forward and participating in your trials.
Well, I would say, for these kind of trials, we have to think about humankind and the challenge of diabetes and what it means to live with it. And while we're looking for the first patients, we're not going to cure the very first patients that we do. They're going to be, if I can use this word, like guinea pigs, essentially, where we will essentially test the safety of the therapy. And it's safety. You're putting very small amounts of cells in. We're not trying at the beginning to get a home run. And once we show it's safe, then we can put our foot on the gas pedal and then put more cells in and allow it to be more therapeutic. But at the beginning, we won't be able to move forward unless we have those first few patients. They're so important to us.
I have another question here from Emma Faith. She says, are these cells protected against the same attack from the immune system that led my baby to type 1 diabetes in the first place?
That's a really great question, and I don't know the answer. I will know the answer when the first patients are treated, because we'll take patients with type 1 diabetes, the autoimmune process that you mentioned there, and when we transplant these cells without any anti-rejection therapies, we'll take samples of those cells at different time points, and we'll look under the microscope and see if the cells are being attacked, see if they survive, and we'll know right away whether or not the gene edits that have been carried out, the 2 knock-ins and the 2 knockouts, whether or not they've been successful at stopping that exact process. And we're hopeful that it will, but we have to do the trial and test and prove that's the case.
Incredible. A great question as well from Rick Tiedemann. He says, are the inclusion or exclusion criteria for the trial participants?
Okay, well, for this trial, because we're not using anti-rejection drugs, we don't have to pick the worst of the worst patients with type 1 diabetes. We need patients with stable sugar control, but we're not looking for the worst of the worst. In the islet cell transplant program that we've been running for the last 20 years, we have been looking at those extreme patients that really have troublesome lows in blood sugar. In this particular trial, we're not looking for that. We're looking for some volunteers that have stable type 1 diabetes, that are adults at this point, and are willing to undergo small superficial surgical implant and explant procedures so we can test these cells.
A question from Tori Oliver. Does cord blood benefit this whole process at all?
Well, it may. So cord blood is rather like the stem cells that we're talking about in the blood. We're not working on cord blood because not everybody has a cord blood sample saved up. And we just thought it would be simpler for everybody if we could develop this process by using patients' whole blood. But it might be possible also to use the same kind of process with cord blood. And we need to think about that. It's a good suggestion.
Shelley asks, who will you take for trials? My daughter is brittle, but now lives in Victoria.
Yeah. So, well, it depends on the trial. As we move forward with that first ViSight trial with these gene edited, they're called PET-QT cells. When we start with that trial, we're probably going to look for patients locally in Edmonton to start with, because it will be complex for a patient to come back and forth from Victoria or elsewhere to have the procedure and then be monitored. I mean, it's possible, but we prefer, I think, for the first few patients at least to have somebody locally. But as we become more effective with this and show that it works, then I think we will be looking across the country and maybe internationally for suitable patients that would fit the trial.
So exciting on that note. Ralph Brooke Holmes asks, why are the IPCs injected into the liver instead of the pancreas? And why does one's immune system not recognize the new IPCs if they came from the individual's own stem cells?
Okay, well, that's a really key question. Difficult to answer to a general audience, but Why do we put them in the liver? Well, we won't start by putting them in the liver, just to be clear. We'll be putting them to begin under the skin. They're basically samples. We call them sentinels. So they basically will act as a marker to see how successful we are with the immune system. But as we learn more and as we want to put more cells in, then we'll move to this very successful site for us, the liver site, the intraportal site inside the liver, because it has a good blood supply and the cells are able to survive better in the liver than they are under the skin. But of course, it's much easier to remove a small sample from under the skin than it is to remove cells that are put into the liver. So we want to be sure first that it's safe.
Simon asks, what are the surgical procedures you talk about when injecting the new cells?
Okay, well, the first trials will involve tiny incisions, about a centimeter or less on the forearm and on the tummy wall, and we'll thread these cells that'll be held in tiny little postage stamp devices under the skin, and we'll take them out, different ones at different time points, and look under the microscope.
So the smallest of incisions.
Small, very small, yeah.
We're going to talk a little bit more about that later when we talk with Kerry, one of our patients, and he's going to try and show you the incision, but I don't even think we can see it at this point. So many questions for you, Dr. Shapiro. Rocco wants to know, what's the cost to get this done?
Well, at this point, it's experimental, so all the costs are covered. But the cost of the, if we're talking about the patient, this trial with ViaCyte, the costs are going to be covered by that company. If we're talking about the patient's own cells that we're making in the lab right now that Nadish just showed you, that's not covered. That's expensive therapy right now. So we're hoping, as we move this forward, to move a therapy that might cost around $1 million a patient when we went for the first few patients to do this, down to a cost of around $100,000 or $50,000 or maybe $20,000 per patient as we begin to expand and automate and learn more about it. That's the sort of the natural progress of any therapy. If we look at some of the newer therapies that are, say, used in children's blood cancers, so these CAR T cells, when they were first used in patients just a few years ago, they cost $1 million a patient. Now, a university lab can make these for around $20,000 a patient. So I think the same kind of process will happen with this.
Here's the $1 million question. Why does the government not fund this?
The government funds tried, proven science. And at this point, this is still experimental. What we need to do in these first trials is prove that this can work. And as we do that, then people will believe in us more, and we'll get additional grants and investment to get us over to the next stage. And eventually, we hope that industry will take over this and will cover all of the costs, and that we won't have to worry about raising funds. And that'll be a really great day. But for now, we're stuck. We need to do the science. We need to prove this idea can work. And we need the money to be able to do that, make it happen.
A nice comment from Unthinkable Legends says, "Go, Dr. Shapiro, go. This is beyond exciting. Thank you for everything you and your team are doing to get to this place. So much gratitude." That's got to feel good to hear that, doesn't it?
Thank you. Yeah. but we can't rest. This hasn't happened until it happens, and we've got to prove it. And so that's why we're so determined to move forward.
Here's a great question as well from Silverthorn. Can non-diabetics donate blood too as a control?
Potentially. We're actually going to look at different kinds of patients with diabetes. So we're looking at patients with type 1 diabetes. And one of your questions there relates to this autoimmunity in type 1 diabetes. But the vast majority of people with diabetes, living with diabetes today, have type 2, which is not an autoimmune process, but still can be corrected by cell transplant. And we also have some patients that have already volunteered their blood for us to test. And these are patients that have undergone surgery with us for other conditions in the pancreas, and they don't have a pancreas anymore. The pancreas has been removed. And for them, we're also trying to make cells where there's no immune system worries. And these all have to be tested. They're essentially control patients for us.
Aaron Barr wants to know any idea when you might be testing this on patients under the age of 18?
Well, we are allowed, as of in fact a few days ago, we're allowed to test blood from volunteers in the age group 12 to 18. Now, that doesn't mean that we'll be transplanting those cells first off into those patients, but that will allow us to understand a little bit better how well the immune system functions and how well these cells can regenerate and be made from the blood. We think it's probably going to be easier to make cells from a young person than from an older person, but we don't know that yet, and we have to just keep going with this. But the answer to your question is we can do that now, but we won't be entering those children into the first trials, because naturally with any trial that's designed first as safety, we have to be sure it's the right treatment and safe first in an adult before we'll take on a child.
Kimmy Morrell says, you mentioned gene editing. Can you expand on that in relation to this process? This is exciting stuff.
Yeah, so the trials that you're going to hear from Kerry in a little bit, and I don't want to scoop him, but the cells that we put into Kerry were called PECO1, and those are the number one because they were the first cells that were tested. And now those same cells that Kerry received have had these 4 gene edits, 2 knock-ins and 2 knockouts, and they're designed to make the cells that they will be non-inflammatory, so they produce a molecule that is non-inflammatory, and they also have their markers on the surface altered, so they won't be recognized by the immune system. It's really exciting, really novel, and I really hope and very hopeful that it will work.
We're almost out of time for questions. We have a few to close with. Evan Linderman says, what adverse effects do you think could occur in patients?
Well, I think the big worry for us, and one of the reasons why we need to really intensify our research right now on the lab bench, is that we could, as we develop these cells, we've got to be sure that they stay as the islet cells that we want, and that there's no unwanted cells, because we're massively expanding these cells from the blood cells to these iPS cells and then back into the islet cells. And what we want to be sure is that we don't have any what we call off-target cells that could cause problems. And that's what we're really working on right now in the lab, cleaning these cells up, making sure there's no contaminants in them that could cause harm.
I'm going to give the final question tonight to you from Shelley. She says, this is a lot to take in. So in summary, where are we insofar as timelines? How far can we go with current funding? What are the other funding options? And what are the worst case scenarios?
Okay, well, the worst case scenario is that a year from now, I have to close the door, switch the lights off in the lab, and let everybody go. And that would be absolutely tragic. I hope that doesn't happen. I don't think it will. But that's where we're at. That would be worst case scenario. Best case scenario is that we are able to raise the funds that we need through DRIFCan and through the events like this. And then we get through the next few years, and we then secure some major grant funding to take this forward to larger scale trials in patients, where it's proven, where there's a lot more enthusiasm, and where we can get industrial partners as part of this to invest in this and move it forward.
Closing thoughts that you want to leave everybody with tonight, Dr. Shapiro?
We are close. We're very close. But in order to get this over the finish line, we've got a lot of work ahead of us. And we're determined to do it our end, but we can't do it without funding.
Dr. Shapiro, it has been such a pleasure and an honor to have this conversation with you and to facilitate it on behalf of our viewing audience tonight. And I think I can say this on behalf of everyone watching. The world needs you, and it needs your team, and it needs your work. And we are all so grateful for everything that you and your team are pouring into the mission to find a cure. We support you in this journey, and we're going to do everything possible tonight to raise funds. Thank you so much for being so generous with your time tonight, for asking or answering the questions, and for sharing your journey with us. We appreciate you.
Thank you, Carrie. Thanks for doing this. It's amazing.
Thank you so much. Well, friends, how was that for a conversation with one of the leading researchers in the world? It's not very often that A, you get to go inside his lab, his team's lab, and see what they're up to, and B, to understand where he is in this research and this journey and how close they are to finding a cure. But as we've been saying so far this evening, all night, we need funds to keep the lights on in those labs. Like Dr. Shapiro mentioned, a year at best. We've got to keep going. We need your help. If you know people who can help, if you can help, go to the website, DRIFCan.com. Follow the simple steps. It's as easy as possible to make a donation tonight. Keep in mind a couple of things. You can make your donation tonight in the denomination of where you live in. So whatever currency is in the area that you live in, make it in that currency. Also tonight, whatever you donate, you will get a tax receipt for it. And I mentioned at the top of our broadcast that an anonymous donor has come forward and will match up to $500,000 in donations for the month of November. Let's not take the whole month. Let's do it in this hour. We know we can. We know that we're a generous community. And looking at all of your comments, I know how much you want to see a cure come to fruition. So many of you that are watching are either diabetic patients or have children or know people who are suffering. Let's make this happen tonight. DRIFCan.com is where you can make a donation. Friends, we're going to move ahead now and talk to some of the patients who are joining us tonight. It's now my pleasure to welcome Kathleen Gibson, who is a registered dietitian and a certified diabetes educator. She was diagnosed with diabetes when she was just 27 years old, and she does say that she remembers what life was like without diabetes. She has known Dr. Shapiro and his team for quite some time and has undergone 3 islet cell transplants over the past decade. And with that, friends, please join me in welcoming Kathleen Gibson to the broadcast tonight. It is such a pleasure to have you here with us this evening.
Thank you, Carrie. It's a real pleasure to be here. It's quite exciting to hear the advances that Dr. Shapiro has and to participate.
I want you to go back a little bit and talk about how your life has changed once you were diagnosed with diabetes and what life was like before, and then how it's changed to what you're living with now.
So I was diagnosed older. I was 27. Most people are diagnosed in that preteen age. And so I had already gone through adolescence and young adulthood. And so it came as quite a shock. When I was diagnosed, I didn't know or didn't think I knew anybody that had diabetes. And so there was a lot to learn. I was already a healthy person, but there's still something different about suddenly having to take needles and paying attention to everything you eat. And I've always suffered from low blood sugars. So I would always have to have juice boxes stashed everywhere, candy stashed everywhere. All of my coworkers would have to have things just in case, out of the blue, a low blood sugar would hit me. And so that was the main reason why I was a candidate for the islet cell transplant, because I would have 3 or 4 low blood sugars every day in the middle of the day, in the middle of the night.
And I'm sure, Kathleen, so many people who are watching tonight can relate to what you were living through. So you said you underwent your first transplant, the islet cell transplant, in 2012. So how did you feel going into it? And were you nervous at all or scared? Take us back to that time.
I was nervous and scared. About this time 10 years ago, I was going through the workup to get my first transplant with all of the lab work, assessing your liver, assessing your health, assessing your kidneys. And so there's a lot of energy that goes into some of that pre-work. And then you get the call that you're on the list, and it's so exciting. And then you get the call, they're ready for you. And so you just pack up everything and you go to the hospital, and you hardly know what's going on. But it was very exciting and nervous, but exciting, more exciting than not, the promise of having a different life with diabetes.
You didn't hesitate being a part of this research program, because I know so many people that are watching want to get involved.
Not at all. And in fact, my first transplant was not the standard Edmonton protocol. My first transplant was using an experimental immunosuppression drug. And we need people doing research, because you can't study a type 1 diabetes medication or a process on people who don't have the disorder. You need to get a sense of what's actually going to happen in the real world. And so I was happy to be part of this experimental protocol for my first transplant. Yeah.
Okay, Kathleen, talk about how that first islet cell transplant changed and impacted your life.
It was very exciting. About 3 weeks after the transplant, I was able to go off insulin and not have any low blood sugars. It was like a revelation not to have to worry. I think for me, if I think about the biggest shift in my life with diabetes, it's the lack of worry. I still occasionally have some lows, but they're nothing like what I used to have. I'm able to feel them and treat them and move on with my day. And I don't have to be so stressed out about doing activities and wondering, do I have enough supplies with me? Do my friends have enough supplies with me? Have I planned enough in advance to be in a safe place at all times? And so,
That's incredible freedom in your life suddenly.
definitely.
But you did have a second and a third transplant. Why did that happen? Tell us about it.
Well, as I mentioned, the first transplant was using an experimental immunosuppression agent. When you get down to sort of the maintenance dose of the medication, it wasn't enough to protect my islet cells from myself. So that first transplant failed, and then I had to get back on the list. And so then that was the reason for the second transplant. And the second transplant was the standard Edmonton protocol. And I went along for about 3.5 years without needing any insulin. Yes, taking immunosuppression medication every day, but I've been lucky. I don't really have too many complications from that medication. And then I got the flu in February of 2016. And it took out some of my islet cells. And so then I was offered a third transplant, which I jumped at the opportunity. And that one was also the standard Edmonton protocol.
Okay. So that final transplant was some 5.5 years ago, but I understand that you still take insulin and still on anti-rejection drugs. Why is that?
The anti-rejection drugs will protect my body should I need any other type of transplant in the future. And the reality is I'm a postmenopausal woman, and that's a whole nother world just in health. For those of you that aren't postmenopausal women, hold on, buckle up. So the reality is, your body naturally ages and is doing things. And so as my body went through menopause, I needed some insulin to help out the islet cells. I still have functioning islet cells.
Mhm.
I just need a little help from insulin now. So I will always take immunosuppression drugs.
But the journey where you are now compared to where you were years ago is dramatically different how you're living your life, correct?
It is. And I think if I had to sum it up in one word, it would be freedom.
It's a beautiful way of putting it. So on that note, as a diabetes educator, what is your hope for patients? Because you know and remember a life without diabetes, and you now have a life of a lot more freedom. What's your message to those watching tonight?
I think it's get involved in a way that makes sense to you. If you have the finances to donate money to the cure, donate money to the cure. If you are able to participate in the research, participate in the research. But the reality is we are close, and it would be wonderful to have a cure. And as Dr. Shapiro said, not just for people living with type 1 diabetes, but people who live with any type of diabetes.
Mhm.
And that's a lot of people.
A lot.
Who have constraints on their life because of a condition.
Kathleen, thank you so much for joining us tonight, for educating us about your journey and sharing your perspective with us. It helps so much for anyone considering getting involved or living with diabetes to hear a perspective like yours. We appreciate the time that you shared with us tonight and the honesty about your journey. Thank you for being with us this evening.
Thank you, Carrie.
Take good care, Kathleen. Wow, that is some powerful, powerful perspective from Kathleen tonight. And we have another patient we're going to get to momentarily, but it's amazing the comments that I'm watching come in as the conversation continues tonight. Lee Matson says the NIH has $500,000 available for non-US entities to apply for funding for this exact type of trial. I hope they've applied for this, and if not, I hope someone sees it. Well, everybody's now seen your comment, Lee, and I know there are people from DRIFCan that are watching right now. I'm seeing Dr. Shapiro shake his head yes in the virtual green room, so I'm going to safely assume that you have applied for that funding. He says yes. I'm just not sure how that ended up. So I'll wait. Dr. Shapiro, maybe even if you can put your comments in, or I can even bring you back at the end of the broadcast if we have time, because everybody loved hearing from you. We'll circle back at the end, Dr. Shapiro. Why not? Sarah Simone says, go Kathleen. Ralph Brookholm says, excellent comments. Lee, your message should definitely get to DRIFCan. Thank you very much. Ralph, it just did, and we are going to follow up with that for you. He also says, just an FYI, that I notified the Juvenile Diabetes Cure Alliance of this event and hope they will spread the word. JDCA advises how diabetes fundraising organizations are using funds to find a cure. Thanks for that perspective, Ralph. And from Wade Izzard says, thank you, Dr. Shapiro. Sarah says, hi, Lee. DRIFCan has it on our list of grants. So there is the answer. And Lee says, awesome, Sarah. Thank you for the response. Thank you all for continuing to join in the conversation. I know many of you know each other, and it's this type of conversation that helps get the answers that you need tonight. So we appreciate it. Keep it coming. So on that note, let's move on with our second patient tonight. It's now my pleasure to bring Kerry Elliott into the conversation. Kerry works with Bell Media as an account executive out of Calgary, who has battled type 1 diabetes for the majority of his life. Kerry is the first patient in Canada to undergo the ViaCyte human stem cell transplant, and to date has undergone 5 procedures related to this work, and that he is going to talk about further tonight. So joining us from Calgary, don't judge him because he's from Calgary. I'm just joking. I had to throw that in. Kerry Elliott, welcome so much to our live broadcast tonight.
For the record, you can judge me. I am from Edmonton originally and was
right.
living in Edmonton when I was part of the trial. So you can judge me now that I'm an Edmonton trader.
I think we did actually work in the same building at some point. Our paths didn't cross, but with Bell Media, that's likely. So it's so great to have you with us tonight. I'm sure that you're enjoying the amazing questions that are coming in. Is the conversation not rich?
That's absolutely fantastic. And I have to say, there's stuff I'm learning tonight watching Dr. Shapiro, and that's always the case. I always love talking to him and his team members because they always have so much exciting things happening and moving forward. And that's why I was so thrilled to get to play a role in the study. It was very, very exciting.
Well, you have certainly had an interesting journey with diabetes. 39 years, I understand, since you were first diagnosed. You were just 15 years old. And so that's quite a journey. Tell us what it's been like. What's the road been like for you?
I should start by saying I always like to think I'm like a senior type 1 diabetic. I'm way up there. But I saw there was a bunch of people who have me beat by quite a bit. So well done you, because it's not an easy path. Those of you at 45 and 50 years, congratulations. And even at 2 years, it can be tough. But when I was first diagnosed with diabetes, I was 15, so I was living at home. And this week is my diaversary. Believe it or not, I know it's coincidental, but I remember getting all my Halloween candies and feeling really rough while I was out because I'd gotten over the flu, and then started to feel much worse over the next few days. And sure enough, ta-da! But we went in, and I mentioned this to you previously, that when you're living at home and you have a really great support structure, it doesn't really seem like you have diabetes until you move out of your house. My mom and my dad had type 1 diabetes, for lack of a better term. Like they took, they're checking my blood sugar constantly. They're counting the carbs. They're checking on me. They're waking me up. They're making sure I have my arrowroot cookies and my juice box for school, which my friends would desperately try to steal from me, but you have to hide in your locker. It wasn't until you move out that you realize that, wow, this is really, really an intense disease and quite terrifying, really. And I think I know a lot of the people on here right now watching understand diabetes quite well, but I think there's a lot of people who don't. still. And one of the big things people don't understand is without insulin, our blood sugar goes high. And eventually that will kill us. It wouldn't happen immediately. I could stop taking insulin and I could go for quite some time. I would just get ideally sicker and sicker, and then I would just stop being around. Insulin allows us to control that. But the problem with insulin is if we misshoot, if we overshoot too much insulin, or we miss a meal, or we get caught up in something, we burn off more calories, your blood sugar can drop drastically. And that can put you in the hospital within a couple hours, or can be even fatal in that amount of time. And so in the back of your head, you're constantly thinking, where's my sugar at? Am I dropping right now? And in fact, just before I came out to speak with you, I quickly checked my blood sugar, because the butterflies I get in my stomach before I publicly speak, you get the cold sweat, you get kind of like that little bit of a shaky hands, you feel the butterflies, is exactly what low blood sugar feels like to me. I'm like, okay, am I going low, or am I just nervous? Fortunately, just nervous. But,
That's incredible. Cindy says completely relate to where you are on your own. So I'm sure a lot of people are feeling exactly that same way. And such interesting perspective to think like the disease itself is dangerous, but insulin can be way more dangerous. So looking back on all those years, do you ever think about it? Like, how many injections do you think you've given yourself?
You know, I did the approximate math, and it's probably right around 70,000. So you got to think every time we eat, we would take an injection. And then we're also taking injections, sometimes if you need to correct, and then right before bed, you do the one that lasts all night, rather 24 hours. So originally it was, we take little chunks and you'd have to eat in the exact time and that sort of thing. Now it's a bit more flexibility, but still, so it's a lot of injections.
It's a lot. Incredible. So that leads us into you being the first human stem cell recipient in Canada, which is huge. So how and why did you decide to do this?
Well, I was fortunate. A family member reached out to me and said, hey, Dr. Shapiro is doing this study with stem cells. And I said, no, that's the islet cell transplant. And I looked into that, but it's the good news, bad news. It's, Carrie, you're just too darn healthy to take part in this study. It's like, okay, I guess that's good news, but also kind of a bummer. But and so I reached out to them and found out it was actually a new study, a stem cell study. And I contacted them, and within 24 hours, they got back to me. It was very, very fast. And so they asked me a series of questions just about health. And I've been very fortunate that besides type 1 diabetes, I've been a fairly healthy guy. So I didn't have any like major conditions that would like eliminate me from the study. And so it's fast tracked within a couple of months. I don't even think 2 full months. It would have been June, and by the first week of August, I was heading in to have the procedure done.
So did you know that it was going to help you, or what was the answer? You're heading in to get this procedure done, and then what?
Well, Dr. Shapiro and his team made it clear that and he mentioned this earlier, the first people in aren't about being cured. The first people in are to see the efficacy and safety. Do the stem cells survive? And is it safe for me? Is it safe for diabetics to move forward? Because you can try things in the lab as many times as you want, but the human body is a very, very complex thing. Like Kathleen talking about menopause, completely changed things for her too, right? And every human's a bit different. So how will they react in humans is very different than how they react in the lab, which is why it's so important to get volunteers. And they did make it clear that we're not putting enough stem cells in to cure you. You're not going to be off insulin. That's not what this is about. This is about finding, is it safe, and does it work, and do we need to pivot? Do we need to shift our direction a little bit to keep going forward with it?
But I have to ask this question. If you knew it wasn't going to help you, it's a big step forward to participate in something like this. Why'd you do it?
Well, I have to be honest with you. It's a little self-serving because eventually if they get to a cure, I like to say, yay, I did a tiny, tiny piece. But more importantly, I think at the time when I talked to the team about it, every type 1 diabetic, every diabetic fights a battle with diabetes every single day, and all we can do is fight it to a standstill. It's just a battle. I'm not a scientist. I'm not a physician. I can't actually take the fight to diabetes. So when Dr. Shapiro and his team came to me and said, this is your opportunity to do that, you can take the fight right to diabetes and help take the first step to lead us towards the next step, and then ideally a cure for diabetes. And even if I played a tiny, tiny role in moving this forward, I couldn't say no. I really wanted to be part of it. Come
Amen
on,
to
you're
that,
killing me.
Carrie.
No, no.
Amen to that. Yes, because that's what we need to be doing, because that's like an honor and a privilege to be a part of this. And Dr. Shapiro was talking about it earlier, how these cells are implanted. So do you have scars? Can you show us where and how big the it was the implants into you?
Well, I can tell you right now. I'll show you the one on my arm because I think it's probably better I keep my pants on for this interview because
yeah, probably.
it's family friendly. So let's keep it safe. So I don't even know if you can see. There's a tiny right here incision just below my finger there. Yeah, you probably can't. I can barely see it, and I'm in my own room. And so what they did is they took 6 postage stamp size devices, which are paper thin, half saturated with stem cells and then half not, just to see how they react in the body. And 2 were implanted in my forearm, and 4 were implanted in my stomach, my abdomen. And then 2 larger ones would have been about the size if you took a credit card and cut it long ways down the middle, but much thinner and was saturated with stem cells, were put in they call it the flank, which I always kind of find funny, but they put it in the flank. And those ones were to stay the entire duration of the 2-year study. And so they went in and put them all in at the same time. And then over the course of just a few weeks, they went in and took out the postage-sized stamps to see, are the stem cells surviving? How's my body reacting to those and to the devices they're in? Then they went the ones in the abdomen, and then the next 2 in the abdomen, and then the remaining 2 stayed in my body for the entire duration, just to see what would happen to them. And again, it wasn't about curing diabetes. It was to find out, are we on the right path? And is it safe, or do we need to pivot?
Okay. So obviously we need to get this across the finish line. How do we do that, Carrie?
Well, and we've talked about it a lot, and I know we've really driven this home, and everyone watching, I think, understands this, but the 2 pillars, really, the research team is at it, Shapiro's at it, his entire crew, and I know them, and they're just a fantastic group of people to work with. They really care about the study and the people in it, but it's finance, and it's subjects, it's people volunteering, and then it's money, and without taking that equal step up, you wind up, okay, well, finance gets too high, or we have patients lined up, but we don't have enough money, like Dr. Shapiro said, could potentially run out in a year, and that's terrifying. So if you are in a position to be in the study and you think it's a fit, I would absolutely recommend. Plus, you get to have you get to do cool interviews like this. How awesome is that? Hang out with Carrie and Shapiro. That's fantastic. But you're also pushing it forward. You can play a bigger role than just fighting diabetes. The other side, if you have the funds to donate, please do. It's really what we need to keep this moving forward. It'd be really, worst case scenario the person asked would be if this just fell off the cliff because of funding. So if anyone's in a position to do that, I really strongly urge you to do that.
Carrie, what's life like for you today, and what's your final message to everybody watching?
My life right now is it's pretty good. My diabetes didn't change at all as a result of the study. I had the devices in me. It didn't get worse, didn't get better. I have a bunch of new friends that work in the medical community, so that's pretty cool. People I can hang out with on a hit Edmonton. But yeah, again, the take home is if you're in a position to help fund it or be part of the volunteering, please do.
I have a few comments to close tonight. Rick Tiedemann says, love your passion, Carrie, and you are a great ambassador for getting involved. Joel says, really appreciate your involvement, Carrie. Ralph says, way to go, Carrie. You are a courageous pioneer. Wow. That is a beautiful way to frame it. I'm going to give the last word to Ralph with you. You are a pioneer, Carrie, and we need people like you to help Dr. Shapiro and his team bring this across the finish line. Thank you so much for joining our live broadcast tonight and for sharing your journey and for your experience to date. I know that you're an inspiration to so many that are watching tonight.
Well, thanks. I'm just excited to be here. Thanks so much, Carrie.
Thanks, Carrie. Wow, great perspective, talking about bringing it across the finish line, and of course the need for funds and the need for volunteers. And again, if you are looking to make a donation tonight, it's easy. Just go to the website DRIFCan.com and just follow the steps there. You can make it in the currency where you live. You also get a tax receipt for all of your donations. And keep in mind as well that an anonymous donor has come forward and pledged $500,000 to match up to $500,000 all donations that are coming in. So let's maximize it. Let's maximize all of that money and make it happen. I do also have a request tonight from the DRIFCan team. They are asking that you go ahead and follow their social media as well. They post a lot of updates on there. So all you have to do is go to DRIFCan social media. We'll put up the handles later, but just remember, there it is right now. So DRIFCan on Facebook and at DRIFCan. So DRIFCAN on Instagram as well as on Facebook. So give them, and they're also on Twitter. They're on Twitter. It's at DRIFCan or at DRIF_can. So you can find them on Twitter. You can find them on Facebook, and you can find them on Instagram. And look at this, we even have them on LinkedIn as well. Diabetes Research Institute Foundation Canada, commonly known as DRIFCan. So that's how you can stay in touch and up to date on all of the latest developments with Dr. Shapiro and his team, as well as with the latest developments in funding, because we know we need to get this across the line. So with that, why don't we go ahead for further perspective on research and ensure that this gets to a point that we can call it a cure? I am thrilled to welcome Rod Otway to the broadcast. He is the chair of the DRIFCan board and also CEO of Executive Vice President, Bird Construction. He is joining us from Edmonton tonight, and you are still in your office.
I am, Carrie.
How have you enjoyed this broadcast so far? I know that you are the chair of DRIFCan. It must really fill your bucket tonight to see this type of support and to see this type of conversation taking place.
Absolutely fantastic. And I know that I'm in the queue to be part of this tonight. And so you, like Carrie said, you have a few butterflies, but halfway through, I just kind of forgot and just watched the show. And like Carrie and others have said, I've been learning so much about diabetes and how people live with it, and they're so courageous in terms of it's part of their everyday life, and they just they carry on. And just so honored to be a small part of it, to help move along on the research side.
You know what's so amazing about what you said, Rob, and I do appreciate that you've learned so much. So here you are, the board chair of DRIFCan. You are without a doubt a leader in our business community, but you don't have a genetic tie or reason, like a personal or family reason to support diabetes. So why is this organization and this research so important to you?
Well, it was sort of a little bit of chance of how I got involved in it. I got a call from our switchboard at work that there was a gentleman that wanted to talk to somebody in charge, and I said, "You got to be kidding! There's probably 30 vice presidents here. Why did they call me?" And turns out he was a former superintendent and who was associated with the DRIFCan board, and asked if I could meet a young lady named Melanie Hibbert, who's our executive director, and if anyone's met Melanie, she's got lots of energy and two children now in their 20s that have type 1.
Yep.
I said, of course I'll talk to her, but I'm not quite sure what exactly as a construction person I can do to help. But as it turns out, you start to talk to them and said, well, I can connect you with a few people I know in the community.
Mm-hmm.
And that led to more discussions and strategies about how can we raise some money? Because to me, that if we had to, we're shooting to raise about 25 million in round numbers, and you saw the numbers earlier there from Dr. Shapiro as sort of the next phase, and 14 billion. I had no idea that was how much it costs, Canadians. And that's
incredible.
Crazy.
It really is. And, on that note, when you're listening and you're learning, you were truly moved by the work that Dr. Shapiro and his team are doing, right here in Edmonton. We are this close to finding a cure. So was that the why behind your involvement?
I think, from a business perspective, and I've been to many, many fundraisers over the years, and we call them the rubber chicken dinners after a while because you eat a lot of chicken.
Yes.
And the penny kind of dropped for me after going to some similar events for many years, and it said, okay, this year we're gonna really move the needle. And I said, wait a minute, we said that 4, 5, 6 years ago, and didn't feel like things were moving that fast. So I really wanted to focus for my own personal contributions on more cure-based organizations versus treatment. And I know we need treatment, obviously, and it seems to me that there's a disproportionate amount of money that goes to treatment versus actually, financing a cure. So
Mhm.
the engineering brain in me sort of kicked into gear when I had a chance to meet Dr. Shapiro and go to one of his lectures. And I just said, I got to do something here to help. So I joined the board. And what I do is, I guess, I bring the business side to the board to help organize. It's been a very grassroots organization. And through that, you get to meet some and hear some amazing stories from the broader community. But we need to raise some serious cash here. And so we're revamping the board a bit. We've created an advisory board of local Edmonton-based heavy hitters in the community that are going to help us, connect with people
Mhm.
that are able to donate, and also perhaps come up with some different events. And as you can appreciate with COVID, that has been a real hard hit to the entire nonprofit community, and especially if you're doing a golf tournament or some type of event where people need to show up in person. So we did this last year as a start, and it was a great hit in terms of awareness, but not really great on the fundraising side. And it's one aspect of a multi-pronged approach that we're taking to increase our donor base, and equally as important, the awareness piece. So, tonight we're being, with the help of you, we're telling stories, and
Mhm.
we're hearing about all the incredible progress that's being made. It's being recorded. It'll be on our website,
Yes.
and people can do a small part by just sharing it with other people they know. And
Sharing, education, awareness, it's all part of it. But like you
yes,
said, we need to raise some serious funds. So we have
yeah,
Will Hergott coming forward with a
yes.
$25,000
Thank you,
donation
Will.
tonight. Thank you, Will.
Thank
And
you.
of course, an anonymous donor saying that they're going to match up to
Yeah,
$500,000 in donations. Like this is huge money, Rob. You must be
fantastic.
thrilled with this. How does it make you feel?
It's well, it's very humbling, really. Like it makes me feel very small in the grand scheme of things. And so thank you to Will, and thank you to our anonymous donor. Thank you to our regular donors, and we have a broad base of people that contribute, and a very small portion that contributes significantly. So thank you to all of you, and I'm sure some of you are out there are part of our regular donors. So I wanted to say thank you to you for advancing and keeping the lights on for James. As he was talking, it reminded me we're like a startup, and we're in the very early stages of proof of concept of a cure. And I'm a big fan of Tesla, and it's kind of the same process where they did a small production line of very
Yeah.
expensive cars, proof of concept, and they moved on to something that's more mass produced. And it's kind of the same thing with the first couple patients will be $1 million patients, and then eventually it'll come down to 20,000. And that just takes a lot of time, effort, and of course money. So
And on that note, Rob, I understand too that like you lead, well, I know that you lead by example in our community. And you told me you have some news that you want to share with everybody tonight.
yeah, I think as board chair, I need to set an example in terms of some donations, and so I've committed to a $10,000 donation, personal donation, to DRIFCan.
Like
And
right, like you're seriously making a $10,000 donation
yes,
like right now?
yeah,
Oh my
yeah,
gosh.
it's
Wow.
thank you. It's I mean, obviously we need more, but it's hopefully it's a step forward, and helps others to donate as well. So that's what I can do, my small bit to help out.
Incredible, Rob. There, yeah, like Wow, I am I'm taken aback by it. Thank you so much for everything, for your donation, and for everything that you and your team at DRIFCan are doing to support Dr. Shapiro and his team in as we accelerate the cure for diabetes. We appreciate your perspective, your donation, and for being with us in this live broadcast tonight.
Thank you, Carrie, and thank you for your support. And
It is my pleasure.
Okay.
Wow, what a night tonight, being able to have a tour of an Edmonton lab where all the magic has happened, talk to patients who undergone different procedures, to have Nadish take us into the rooms and explain, and of course, to have the amazing Dr. Shapiro talk about the research and answer questions. We still have time tonight. We still have time before we sign off. So, and I did this on purpose because I know you have more questions. So Dr. Shapiro is still there and I want to bring him back. What do you think? Should we? It's not planned. Yes, everybody says, let's do it. I had to do it, Dr. Shapiro. So many comments, so many questions. As you're watching, thoughts right now?
Well, I'm just so touched by Rob's words, by Kerry and what he went through. I remember the first days when I was trying to, ask him and make sure I didn't twist his arm too much to be part of those trials. It is like a startup, and we'll get beyond where we are right now with your help. We're determined at our end. We need help. We're getting the help, and thank you so much for your generosity. So many of you. We'll do this together, and I'm very, very confident at the end of the day we'll have something far, far better for diabetes treatment than we can ever imagine, and it will be as close to a cure that we'll get.
Dr. Shapiro, I'd love to address a few more questions. Did any come to mind while you were listening? I can go back in the comments, but I know they're coming in fast and furious. But does anything pop into your head that you heard that you wanted to address?
Yeah, I saw one comment about that, a recent trial that's just gone ahead by a different company called Vertex, where they put cells, like we have done, into the portal vein in the liver with stem cell derived products. They have to take anti-rejection drugs at this point with that trial, but that's also showing really great promise. I think that first patient is off insulin that's been treated in that way, and it really shows that across the field there's so much happening in this space, and we're part of it. We're going to drive things forward as best we can from our end, but this is happening now in real time, and there's so much possibility for patients now in the future that we could never have even dreamed about before. So it's happening.
A question from Rebecca Shorey. She says, any idea of when the documentary, The Human Trial, will be released? This was featuring ViaCyte and Dr. Shapiro. Hearing about how close they are getting to a cure gives hope.
Yeah, I don't have an answer to that. I'd have to ask the human trial team directly. It was a while since we began the filming of that, but it'll be a very telling documentary because ViaCyte opened the doors from the very beginning and showed every aspect of that trial development, and not many industries would have been willing to do that at their stage. So I think it'll be a very interesting documentary.
Okay, I'm looking forward to seeing it as well. A lot of questions tonight, and this one is reflective of coming in. Gunley Wesley says, what about clinical trials for type 2 diabetes? Lots of people are asking about type 2.
There's no question that the cell replacement therapy that's been working so well for patients in type 1 diabetes will be so effective too in patients with type 2. No question at all. The way I see it is if we can lower the need for the anti-rejection drugs or hopefully eliminate them altogether, these treatments, these autologous patients' own cells, all these stem cells that have been gene edited will be very, very fitting for patients with type 2. No question about it.
So, Dr. Shapiro, can you give us a brief recap tonight of just exactly where we are for anyone that didn't see the beginning of your talk, to just recap the research that's going on and where you are?
Yeah, put very simply, we believe in cell replacement therapy for patients with all forms of diabetes. So in type 1 diabetes, the patient's own immune system's destroyed their cells. We're going to put new cells back in. In type 2 diabetes, we can replace the need for cells. For the last 20 years, we've been transplanting cells from organ donors. Now we have a limitless source of cells that we can manufacture in the lab using patients' blood. And we now have to show that that is safe and that it can work. And that's what we need to do in the next year or so. Show that it's safe, show that it can work. Then we can escalate the dose up and move from this startup to a real cure for patients with diabetes. But we need your help to do that.
And that help comes in the form of patients. So go ahead and remind everybody.
Yeah, we need patients to sign up for the trial. Please contact the email that's on your screen now, citp@ualberta.ca. If you're a potential patient, you can contact our team. We'll get back to you as promptly as we can, and hopefully you'll be suitable. And we also need your help with fundraising.
Dr. Shapiro, every single time I have the opportunity to sit down with you and have an interview, it's a gift. It's a gift for me to be able to be a recipient of that conversation, but a true gift tonight to have so many people around the world listening to you and learning about the work that you really have dedicated your life to. What a privilege to have you and your team here in Edmonton. And we will be cheering you on and doing everything that we can to get this across the finish line for you. Thank you so much for all you are doing to find a cure for diabetes.
Thank you, Carrie. Amazing evening.
It truly has been. Well, everybody, I hope that you have enjoyed this conversation, enjoyed the perspective that Dr. Shapiro brought to the conversation and the updates that he was able to explain to you about his research. And then, of course, there's Nadish taking us right inside that lab. I have covered Dr. Shapiro's work for probably almost 3 decades, and I've never been inside that lab. So I was so grateful to have a look at what he was doing, and that was a real privilege. So I wanted to say thank you to the team, the islet cell transplant team, and everything that they did to make tonight a reality. I can see Nadish is still in the virtual green room. Thank you, Nadish, for making that happen. I really appreciated that tour, and I know everybody watching tonight. So many thank yous coming in. Thank you all for sharing your time with us tonight. Cindy says thank you to Dr. Shapiro, his team, and all the volunteers. Cindy, I echo your sentiments as well. And as I close out the evening, I want to remind all of you again tonight that if you want to make a donation, all you have to do is go to the website, DRIFCan, DRIFCAN.com. Follow the simple steps. Once again, you can make the donation in the dollars of where you live. So wherever you're joining us from, you can make it in that currency. You will get a tax receipt for it. And remember that an anonymous donor has come forward and said that they will match donations up to $500,000 for the month of November. So everybody watching, let's max that out. A big, huge thank you to Will Hergott for his $25,000 donation. Incredible generosity. Another huge thank you to Rob Otway, the board chair of DRIFCan, for his $10,000 donation, and for every single one of you that donated money tonight and shared your time with us this evening. We appreciate you. We appreciate you coming on this journey with us this evening, for supporting DRIFCan, and ultimately for supporting the work of Dr. James Shapiro and his team. With your help, we will make this happen. Thanks so much for watching, everybody. On behalf of DRIFCan, we appreciate you. Have a fabulous evening. Good night.